CRISPR. Could we edit out aging itself? Close your eyes and feel the gentle rhythm of your heartbeat. Listen to the soft whisper of your breath as it flows in and out, carrying life to every corner of your being. In this moment of stillness, something extraordinary is happening within you, something so subtle, so profound, that it has taken humanity thousands of years to begin to understand it. Deep within your body, in the microscopic universe of your cells, ancient clocks are ticking. Not the mechanical clocks that measure our days and nights, but molecular timekeepers that have been counting the moments of your existence since the very first spark of your life. These are the clocks that govern aging itself, the mysterious process that transforms a newborn's soft skin into the weathered hands of an elder, that changes the boundless energy of youth into the measured wisdom of age. Imagine, if you will, that your body is a vast city, bustling with activity even as you rest. In this cellular metropolis, trillions of inhabitants work tirelessly through the night, each one carrying out its designated purpose. Some are builders, constructing new proteins and repairing damaged structures. Others are messengers, carrying important information from one part of your body to another. Still others are guardians, protecting you from invaders and maintaining the delicate balance that keeps you alive. But what makes this city truly remarkable is that every single one of these cellular citizens carries within itself a clock, a biological timekeeper that determines when to work, when to rest, when to divide, and ultimately, when to fade away. These molecular clocks don't tick like the grandfather clock in your hallway. Instead, they pulse with chemical rhythms, their tempos set by the ancient dance of molecules that began billions of years ago in the primordial soup of early Earth. Your cells age through a process as natural as the changing of seasons, yet far more intricate than any symphony ever composed. Each time one of your cells divides to create new life, it must copy its entire instruction manual, the DNA that contains the blueprint for everything you are. This copying process is remarkably precise, yet it is not perfect. Like a scribe, carefully transcribing an ancient text by candlelight, occasionally a letter is changed, a word is altered, or a sentence is lost. Most of these small changes are harmless, absorbed into the vast redundancy of your genetic code like raindrops into an ocean. But over time, as the years accumulate like layers of sediment in a riverbed, these tiny imperfections begin to add up. Your cellular machinery, once as precise as a Swiss watchmaker's tools, begins to work with less efficiency. The proteins that maintain your tissues start to accumulate small errors. The energy factories within your cells, the mitochondria that power every thought and movement, begin to produce less energy and more waste. This is the gentle, inexorable process of aging unfolding within you right now as you listen to these words. It is not a disease to be cured, but rather the natural consequence of being a complex, living system in a universe governed by entropy, the fundamental principle that all things tend toward disorder over time. Yet even as this process continues, your body performs daily miracles of maintenance and repair that would astonish the greatest engineers. Consider the remarkable fact that most of the atoms in your body right now were not there 7 years ago. You are constantly rebuilding yourself, replacing old cells with new ones in an endless cycle of renewal. Your skin completely replaces itself every few weeks. The lining of your stomach renews itself every few days. Even your bones, which seem so permanent and solid, are constantly being broken down and built back up, refreshed and renewed by specialized cells that work like tireless construction crews. This process of renewal is one of the most beautiful aspects of being alive. You are not a static sculpture, carved once and left unchanged. You are more like a river, always flowing, always moving, always becoming something new while maintaining your essential nature. The water that flows past a particular rock today is not the same water that flowed past yesterday, yet we still call it the same river. But here lies one of the deepest mysteries of life. If you are constantly renewing yourself, constantly replacing your old cells with fresh ones, why do you age at all? Why doesn't this process of renewal keep you forever young? The answer lies in those molecular clocks we spoke of earlier, the genetic timekeepers that ensure each new cell is slightly different from the one that came before. These differences accumulate gradually, like the slow wearing away of a stone, step by countless footsteps. Each generation of cells carries forward the accumulated changes of all the generations before it, along with a few new changes of its own. Over time, these changes affect how well your cells can perform their functions, how efficiently they can repair damage, and how effectively they can communicate with their neighbors. Yet recent scientific discoveries have revealed something that would have seemed like magic to previous generations. These molecular clocks are not immutable. They are not carved in stone, unchangeable and eternal. Instead, they are written in the language of genetics, a language that we are finally beginning to learn to read, and perhaps even to edit. Scientists have discovered that some creatures seem to have mastered the art of resetting their biological clocks. There are jellyfish that can reverse their aging process, essentially becoming young again. There are trees that live for thousands of years, their cellular clocks ticking so slowly that centuries pass like seasons. There are tiny worms that can extend their lifespans dramatically through simple genetic changes. These discoveries have opened a door to possibilities that once existed only in the realm of dreams and stories. What if we could learn to reset our own molecular clocks? What if we could edit the very genes that govern aging, teaching our cells to maintain themselves with the efficiency of youth throughout our entire lives? As you drift deeper into the peaceful realm between waking and sleeping, let your mind wander through the microscopic universe within you. Feel the gentle rhythm of your cellular clocks, ticking away the moments of your existence with their ancient, patient cadence. Marvel at the intricate dance of renewal and aging that makes you who you are, a temporary arrangement of atoms and energy that has somehow learned to contemplate its own existence. Tomorrow, we will explore the language in which these molecular instructions are written, the elegant, spiraling code of DNA that contains the blueprint for everything you are and everything you might become as you settle deeper into the quiet comfort of this moment. Imagine yourself shrinking down, becoming smaller and smaller until you can slip through the walls of your own cells and enter the most magnificent library that has ever existed. This is not a library of books and papers, but something far more extraordinary, a cosmic archive written in the elegant language of DNA, spiraling through the nucleus of every cell in your body like a graceful double helix dance that has been choreographed over billions of years. Here, in this molecular sanctuary, the story of you is written in letters so small that millions of them could fit on the period at the bottom of this sentence. Yet these tiny symbols contain more information than all the libraries of the ancient world combined. They hold the instructions for the color of your eyes, the rhythm of your heartbeat, the way your mind processes these very words, and countless other details that make you uniquely, irreplaceably you. The language of DNA is beautifully simple, yet infinitely complex. It uses an alphabet of just 4 letters, T, G, and C, representing the chemical bases adenine, thymine, guanine, and cytosine. These 4 molecular letters combined in endless variations, like notes in an eternal symphony, spelling out the instructions for life itself. If you could read this genetic text aloud, speaking 1 letter per second, it would take you nearly a century to read through the complete instruction manual contained in just one of your cells. But DNA is not merely a static library gathering dust on forgotten shelves. It is alive with activity, constantly being read, interpreted, and acted upon by the cellular machinery that surrounds it. Imagine molecular readers moving along the spiral staircase of your genetic code, their tiny fingers tracing each letter, translating the ancient language into the proteins that build your body, repair your tissues, and carry out the countless chemical reactions that keep you alive. This process of reading and interpreting your genetic code is like watching the most intricate dance ever performed. Specialized proteins called enzymes unwind the double helix with the gentle precision of a master calligrapher unfurling a scroll. Other proteins slide along the exposed genetic text, reading the sequence of letters and translating them into a complementary language, the language of RNA, which serves as a messenger carrying instructions from the nucleus to the protein-making factories scattered throughout your cell. These protein factories, called ribosomes, receive the genetic messages and begin the delicate work of constructing new proteins, according to the specifications encoded in your DNA. Each protein is folded into a precise 3-dimensional shape that determines its function, like origami sculptures that can catalyze chemical reactions, provide structural support, or carry messages between different parts of your body. What makes this molecular dance even more remarkable is its faithfulness across time. The DNA in your cells today carries the same basic instructions that were present in your very first cell, the fertilized egg from which you grew. As that single cell divided into 2, then 4, then 8, and eventually into the trillions of cells that compose your body today. Each new cell received an almost perfect copy of this genetic library. Yet the dance of DNA is not without its occasional missteps. Sometimes, as the molecular machinery copies your genetic code, a letter is changed, deleted, or inserted in the wrong place. Most of these changes are harmless, like typos in a vast text that don't affect the overall meaning. Some changes might even be beneficial, providing new capabilities or resistances that help you adapt to your environment. But occasionally, these genetic changes can alter the dance in ways that contribute to aging or disease. A single changed letter might cause a protein to fold incorrectly, disrupting its function and creating a cascade of effects throughout your body. Over time, as these changes accumulate, they can affect how efficiently your cells repair themselves, how well they resist damage, and how gracefully they age. For most of human history, we could only observe this genetic dance from the outside, marveling at its complexity without being able to participate directly. We could read the letters of the genetic code, but we couldn't edit them. We could watch the dance, but we couldn't change the choreography. The DNA within our cells seemed as immutable as the stars in the sky, beautiful, complex, but forever, beyond our ability to modify. Then, in laboratories around the world, scientists made a discovery that would change everything. They found that nature herself had already invented molecular tools for editing DNA. Certain bacteria, under attack from viruses, had developed sophisticated systems for cutting and modifying genetic code with extraordinary precision. These natural editors could locate specific sequences of DNA letters, cut them out, and replace them with new instructions. The most remarkable of these natural editors is a system called CRISPR, name that sounds almost musical, like the crisp snap of autumn leaves or the gentle whisper of pages turning in an ancient book. CRISPR operates with a precision that would make the finest surgeon envious, able to locate and modify individual letters in the vast library of your genetic code without disturbing the surrounding text. When imagine having the ability to edit the instruction manual of life itself, to correct the typos that contribute to aging, to rewrite the passages that lead to disease, to enhance the chapters that promote health and longevity. This is the extraordinary possibility that CRISPR has opened before us, like a door to a room we never knew existed. But with this remarkable power comes profound responsibility. The ability to edit the human genetic code is not just a technical achievement, it is a capability that touches the very essence of what it means to be human. When we contemplate editing the genes that influence aging, we are not just considering a medical treatment, we are contemplating the possibility of fundamentally altering the human experience of time itself. As you drift in this liminal space between waking and dreaming, let your mind wander through the spiral pathways of your own genetic code. Feel the ancient dance of DNA unfolding within you, the billions of years of evolutionary wisdom encoded in every cell. Consider the remarkable journey that has brought us to this moment in history, when we stand at the threshold of being able to edit the very book of life. The genetic code that spirals through you right now connects you to every living thing that has ever existed on Earth. The same four-letter alphabet that spells out your uniqueness also writes the stories of the trees outside your window, the birds that sing at dawn, and the bacteria that help digest your food. You are part of an unbroken chain of genetic information that stretches back to the very origin of life itself. Yet now, for the first time in the history of our species, we hold in our hands the tools to become conscious participants in this genetic dance. We have discovered molecular scissors so precise, so powerful, that we might be able to cut away the very mechanisms that cause us to agend in doing so. We stand ready to explore what it truly means to edit the dance of life itself. In the gentle quiet of this moment, as your breathing deepens and your thoughts begin to slow, imagine yourself as a master librarian in the vast genetic library we explored together. You walk through corridors lined with billions of volumes, each one containing the instructions for a different aspect of your being. But tonight, you carry something extraordinary in your hand, a pair of molecular scissors so precise, so elegant, that they can edit the very language of life without damaging a single surrounding letter. These are not ordinary scissors. They move through your cells with the grace of a ballet dancer and the precision of a master surgeon. They are called CRISPR, and they represent one of the most remarkable discoveries in the history of human science, a discovery that began not in gleaming laboratories or prestigious universities, but in the microscopic world of ancient bacteria, fighting for their lives billions of years ago. When Earth was young and life was just beginning to explore the possibilities of existence, bacteria faced a constant threat from viruses that sought to invade their cellular sanctuaries and hijack their genetic machinery. These viral attackers would inject their own DNA into bacterial cells, turning them into factories for producing more viruses. It was a molecular war fought in spaces smaller than the eye can see, with the very survival of life hanging in the balance. But life, in its infinite creativity, found a way to fight back. The bacteria developed a sophisticated defense system, a molecular memory that could remember previous viral attacks and mount a precise counterattack when the same invaders returned. They called this system CRISPR, which stands for Clustered, Regularly Interspaced Short Palindromic, repeats a name that describes the unique structure of genetic sequences that serve as the system's memory bank. Picture this bacterial defense system as the most advanced security network ever devised. When a virus attacks, the bacterial cell captures a small piece of the invader's genetic code and stores it in its CRISPR memory bank, like a wanted poster in a cosmic post office. The next time that same virus attempts an invasion, the bacterial cell recognizes it immediately and dispatches molecular scissors to cut the invader's DNA into harmless fragments. What makes these molecular scissors truly extraordinary is their precision. They don't slash wildly through genetic code like a machete through a jungle. Instead, they move with the delicate accuracy of a calligrapher's brush, guided by a molecular GPS system that can locate one specific sequence of letters among the billions that make up a genome. When they find their target, they make a clean, precise cut, like a librarian carefully removing a single page from a treasured book. The discovery that scientists could harness and reprogram these bacterial scissors to edit any genetic sequence they chose was like finding the master key to the library of life itself. Suddenly, the genetic code that had seemed as immutable as mountains became as editable as text on a computer screen. Letters could be changed, words could be rewritten, entire paragraphs could be deleted or inserted with unprecedented precision. But CRISPR is more than just molecular scissors. It is a complete editing system, as sophisticated as any word processor ever created. It consists of two main components working in perfect harmony: a guide RNA that acts like a GPS navigator, searching through the vast genetic landscape to find the exact target sequence, and a protein called Cas9 that serves as the actual cutting tool, severing the DNA at precisely the right location. The guide RNA moves through your genetic code like a patient detective, comparing each sequence it encounters to the target sequence it has been programmed to find. It slides along the double helix of DNA, reading letter by letter, until it discovers the perfect match. When that moment of recognition occurs, it signals to the Cas9 protein, which then makes its precise cut, opening up the genetic text for editing. What happens next is where the true magic of CRISPR reveals itself. Once the molecular scissors have made their cut, the cell's natural repair mechanisms spring into action. Like a team of master craftspeople ready to heal the wound in the genetic text, scientists can guide this repair process, providing new genetic material to be inserted at the cut site, or simply allowing the cell to repair itself in ways that effectively delete unwanted genetic information. This process happens with a gentleness that seems almost compassionate. The CRISPR system doesn't force its way into cells or violently disrupt their natural processes. Instead, it works with the existing cellular machinery, like a skilled editor collaborating with an author to improve a manuscript. The result is genetic changes that are so precise, so carefully integrated, that they become indistinguishable from the natural variations that occur throughout the genome. In laboratories around the world, scientists have used CRISPR to perform editing feats that would have seemed impossible just a few decades ago. They have corrected genetic defects that cause blindness, allowing people to see for the first time. They have edited the genes of immune cells to help them better fight cancer. They have even begun to explore the possibility of editing the genes involved in aging itself, potentially slowing or reversing the molecular clocks that tick away the years of our lives. The applications for treating age-related diseases are particularly promising. Scientists have discovered that many of the processes that contribute to aging are controlled by specific genes, genetic switches that determine how efficiently our cells repair themselves, how well they resist damage, and how gracefully they age. By editing these genes, we might be able to enhance our cells' natural maintenance and repair systems, keeping them functioning like younger, healthy cells throughout our entire lives. Imagine having the ability to edit the genes that control your telomeres, those protective caps on the ends of your chromosomes that shorten with each cell division. What if we could use CRISPR to activate the enzymes that rebuild these cellular timekeepers, effectively resetting the aging clock in your cells? Or consider the possibility of editing genes that influence how well your mitochondria, the powerhouses of your cells, function as you age. Enhanced mitochondria could provide more energy and produce less cellular waste, potentially slowing the aging process at its most fundamental level. The precision of CRISPR also opens up possibilities for addressing the accumulation of cells, those aging cells that refuse to die and instead linger in your tissues, creating chronic inflammation and contributing to age-related diseases. Scientists are exploring ways to use genetic editing to either remove these problematic cells or reprogram them to function like young, healthy cells once again. But perhaps the most profound aspect of CRISPR is not its technical capabilities, but the way it changes our relationship with our own biology. For the first time in human history, we are not merely passive recipients of our genetic inheritance. We have become active participants in our own evolutionary story, capable of editing the very code that makes us who we are. This power brings with it questions that extend far beyond the realm of science. When we edit our genes to slow aging, are we changing what it means to be human? How do we ensure that these powerful tools are used wisely and equitably? These are the deep, thoughtful questions that accompany any truly transformative technology. As you rest in this peaceful moment, feeling the gentle rhythm of your breath and the quiet beating of your heart, consider the extraordinary fact that, within your cells right now, the ancient dance of DNA continues, carrying the genetic information that makes you uniquely you. And somewhere in laboratories around the world, scientists are learning to dance with this molecular choreography, wielding their precise scissors with the hope of editing away the very mechanisms that cause us to age. The molecular scissors of CRISPR represent more than just a scientific tool. They are a bridge between the genetic past that shaped us and the edited future we might choose to inhabit, where the protective guardians of our chromosomes might be taught to keep us young far longer than nature ever intended. As you sink deeper into the peaceful embrace of rest, let your mind drift to the tips of your chromosomes, where one of life's most elegant timekeeping mechanisms unfolds in microscopic detail. Here at the very edges of your genetic material, tiny guardians called telomeres stand watch like sentinels at the gates of time itself. These remarkable structures, no larger than a few thousand molecules strung together, may hold the key to understanding not just how we age, but how we might one day choose to age differently. Imagine your chromosomes as precious ancient scrolls, containing the most important documents in the library of your life. Like any treasured manuscript, these genetic scrolls need protection from the wear and tear of daily use. The telomeres serve as protective caps, much like the plastic tips on shoelaces that prevent them from fraying and unraveling. Without these molecular guardians, the chromosomes would be vulnerable to damage and fusion, leading to genetic chaos that could threaten the very stability of your cells. But telomeres are more than just protective caps. They are living calendars that record the passage of cellular time. Each time one of your cells divides to create new cells, a process that happens millions of times throughout your body every day, the telomeres grow slightly shorter. This shortening is not accidental or random. It is a precise molecular clock that has been ticking away the moments of your life since you were born. When you were young, your telomeres were long and strong, like brand new shoelaces fresh from the package. With each cell division, they gave up a small portion of themselves to protect the precious genetic information they guard. Over the years, through countless cellular divisions, your telomeres have grown progressively shorter, like candles burning slowly in the windows of time. This process of telomere shortening is one of the most fundamental mechanisms of aging that scientists have discovered. When telomeres become critically short, cells can no longer divide safely. They enter a state called senescence, where they stop dividing but don't die. Instead, they linger in your tissues like old books that can no longer be checked out from the library but continue to take up space on the shelves. These senescent cells don't just sit quietly in your tissues. They actively contribute to aging by secreting inflammatory chemicals that can damage nearby healthy cells. They accumulate over time, creating pockets of chronic inflammation throughout your body. This inflammation contributes to many of the conditions we associate with aging: wrinkled skin, stiff joints, weakened immune systems, and increased susceptibility to age-related diseases. The discovery of telomeres and their role in aging earned scientists the Nobel Prize in Physiology or Medicine, recognizing the profound importance of these cellular timekeepers. But perhaps even more exciting than understanding how telomeres work is the growing realization that their shortening is not inevitable or irreversible. Nature herself has provided us with examples of creatures that seem to have mastered the art of maintaining their telomeres indefinitely. Consider the remarkable case of the immortal jellyfish, Torotopsis dohrnii. This extraordinary creature can reverse its aging process, transforming from a mature adult back into a juvenile form, essentially resetting its biological clock. During this process, its telomeres are rebuilt and refreshed, allowing it to potentially live forever. While we are very different from jellyfish, the basic mechanisms of telomere maintenance are surprisingly similar across all forms of life. Even more intriguing are the discoveries scientists have made about human cells themselves. Most of your cells have limited ability to rebuild their telomeres, but some special cells in your body, like stem cells and certain immune cells, possess an enzyme called telomerase that can add new molecular building blocks to telomeres, effectively resetting their length. This enzyme is like a molecular fountain of youth, capable of turning back the cellular clock and restoring the protective caps to their full length. The existence of telomerase in your body proves that cellular aging is not a fixed, unchangeable process. Your cells already possess the molecular machinery needed to maintain eternal youth. It's simply not active in most of your cells most of the time. This discovery has opened up extraordinary possibilities for using CRISPR and other genetic editing tools to activate telomerase in cells where it normally remains dormant. Scientists are now exploring ways to use genetic editing to carefully control telomerase activity, potentially extending the healthy lifespan of cells throughout your body. This is delicate work, requiring the precision of a master watchmaker. Too little telomerase activity, and cells continue to age normally. Too much, and cells might become immortal in dangerous ways, potentially leading to cancer. The goal is to find the perfect balance: enough telomerase activity to maintain healthy telomerase, but not so much that cells lose their natural controls. Early experiments with genetically modified cells in laboratory settings have shown remarkable results. Cells that have been edited to express controlled amounts of telomerase can divide far more times than normal cells, maintaining their youthful characteristics for much longer periods. These cells show improved function, better resistance to stress, and enhanced ability to repair damage, hallmarks of cellular youth. The implications of these discoveries extend far beyond the laboratory. Imagine if the cells in your skin could maintain their telomeres indefinitely, continuing to produce collagen and elastin with the efficiency of youth. Your skin might remain smooth and elastic for decades longer than it does today. Consider the possibilities for your immune system cells if they could maintain their telomeres and continue dividing and adapting to new threats throughout your entire life, never losing their youthful vigor. Perhaps most exciting of all is the potential for maintaining telomeres in stem cells, those remarkable cells that can transform into any type of cell your body needs. If we could ensure that stem cells maintain their telomeres indefinitely, we might be able to preserve your body's natural repair and regeneration capabilities throughout your entire life, keeping your tissues and organs functioning like those of a much younger person. The research into telomeres and aging is still in its early stages, but the potential applications are breathtaking. Scientists are developing therapies that could activate telomerase temporarily in specific tissues, allowing cells to rebuild their telomeres and reset their biological clocks. Other researchers are exploring ways to deliver telomerase directly to cells, or to use genetic editing to modify the genes that control telomerase production. But as with all powerful technologies, the ability to manipulate telomeres raises important questions about how we want to use these capabilities. If we can reset the cellular clocks that govern aging, how long should human life become? How do we ensure that these treatments are safe and effective? These are the kinds of thoughtful questions that accompany any technology with the potential to fundamentally change human experience. As you drift in this peaceful space between waking and sleeping, feel the gentle presence of your telomeres. Those tiny guardians standing watch at the tips of your chromosomes. They have been faithfully protecting your genetic material since the moment you were conceived, growing shorter with each passing day as they shield your precious DNA from harm. They are the molecular embodiment of time itself, measuring out the days of your life in increments so small they can only be seen with the most powerful microscopes. Yet now, for the first time in human history, we stand at the threshold of being. They are able to tend these molecular guardians, to replenish their strength and perhaps even restore their youth. The fraying edges of forever may not be fraying after all. They may simply be waiting for us to learn how to weave them back together, strong enough to protect the seeds of renewal that lie sleeping in the depths of your cells, in the quietest chambers of your body, hidden in the shadows between organs and nestled in the protective niches of your bones. Something extraordinary waits in patient slumber. These are your stem cells, the sleeping seeds of renewal that carry within themselves the potential to become any type of cell your body might need. They are your personal fountain of youth, flowing not with mythical waters, but with the liquid possibility of endless regeneration. As you rest in this moment of peaceful contemplation, imagine these remarkable cells scattered throughout your being like stars in a cosmic landscape. Each one holds within itself the memory of your earliest existence, when you were nothing more than a single fertilized cell with unlimited potential. Unlike the specialized cells that make up your skin, your muscles, or your brain cells that have chosen their identity and committed to their specific roles, stem cells remain forever young, forever undecided, forever ready to transform into whatever your body needs most. Your stem cells are the master craftspeople of your biological repair system. When you cut your finger, it is stem cells that awaken and begin the intricate process of healing, transforming themselves into new skin cells to close the wound. When your blood needs refreshing, stem cells in your bone marrow continuously generate new red blood cells, white blood cells, and platelets, keeping your circulation vibrant and healthy. When your muscles need repair after exercise, stem cells contribute to the rebuilding process, helping you grow stronger than before. But perhaps the most remarkable thing about your stem cells is their relationship with time itself. While most of your cells age and accumulate damage over the years, stem cells possess an almost magical ability to maintain their youth. They divide slowly and carefully, preserving their pristine genetic material while sending out specialized daughter cells to do the heavy work of daily maintenance and repair. In this way, they remain like eternal springs, always fresh, always ready to flow. However, even these remarkable cells are not completely immune to the passage of time. As you age, your stem cells gradually become less numerous and less active. The vibrant repair mechanisms that kept you resilient in youth begin to slow down, like rivers that once flowed freely but now move with less urgency. Your wounds heal more slowly, your muscles recover less quickly from exertion, and your body's overall capacity for renewal gradually diminishes. This decline in stem cell function is one of the primary reasons why we age. Your body is like a magnificent city that depends on a workforce of repair crews to maintain its buildings, roads, and infrastructure. When these crews are young and numerous, they can keep everything in perfect condition. But as they age and their numbers dwindle, the city begins to show signs of wear and tear. Streets develop potholes, buildings begin to crumble, and the overall vitality of the city slowly fades. Scientists have discovered that much of this decline in stem cell function is controlled by genetic switches, molecular programs that determine when stem cells should be active and when they should remain dormant. As we age, these genetic programs gradually shift toward dormancy, like dimmer switches being slowly turned down, reducing the activity of our cellular repair crews when we need them most. But here lies one of the most exciting frontiers in the science of aging. These genetic switches are not permanent. They can be reset, reprogrammed, and optimized using the precise tools of genetic editing. Scientists have discovered that by carefully modifying specific genes and stem cells, they can restore much of their youthful vigor and activity effectively, turning back the biological clock that governs cellular renewal. Imagine if the stem cells in your muscles could be genetically edited to maintain their youthful enthusiasm throughout your entire life. Your muscles might continue to repair and regenerate with the efficiency of a 20-year-old, regardless of your chronological age. Consider the possibilities if the stem cells that maintain your brain could be enhanced to continue generating new neurons and supporting brain cells, potentially preventing the cognitive decline that often accompanies aging. The research in this area is already showing remarkable promise. Scientists have used genetic editing techniques to rejuvenate aged stem cells in laboratory settings, restoring their ability to divide, migrate, and differentiate into specialized cell types. These rejuvenated stem cells behave like young cells in every measurable way, producing the same proteins, responding to the same signals, and maintaining the same high levels of activity that characterize stem cells in youth. Even more exciting are the discoveries about induced pluripotent stem cells, adult cells that have been genetically reprogrammed to return to a stem cell-like state. This process, which earned its discoverers the Nobel Prize, essentially allows scientists to take any adult cell from your body and reverse its developmental clock, transforming it back into a cell with the unlimited potential of youth. The three reprogrammed cells can then be guided to become any type of cell needed for repair and regeneration. The implications of this technology for treating age-related conditions are profound. Imagine being able to take skin cells from an elderly person, reprogram them back to a stem cell state, and then guide them to become young, healthy heart muscle cells to repair a damaged heart. Or consider the possibility of reprogramming cells to become fresh neurons to replace those lost to neurodegenerative diseases. CRISPR and other genetic editing tools are making these possibilities increasingly practical. Scientists can now edit the genes involved in stem cell programming with unprecedented precision, fine-tuning the molecular switches that control cellular age and identity. They can enhance the genes that promote stem cell survival and activity while silencing the genes that contribute to aging and decline. One particularly promising area of research involves editing the genes that control stem cell metabolism, cellular processes that generate energy and manage waste. Young stem cells have highly efficient metabolic systems that keep them healthy and active. As stem cells age, the metabolism becomes less efficient, leading to the accumulation of cellular waste and reduced energy production. By editing the genes that control these metabolic processes, scientists hope to maintain the energetic vitality of stem cells throughout life. Another exciting frontier involves editing the genes that control how stem cells respond to their environment. Stem cells exist in specialized niches throughout your body, protective microenvironments that provide the signals and nutrients necessary for stem cell health. As we age, these niches can become less supportive, sending signals that encourage stem cells to remain dormant. Genetic editing might allow us to modify stem cells so they remain active and responsive even in aged tissue environments. The research has also revealed fascinating insights about the relationship between stem cells and the overall aging process. Scientists have discovered that maintaining healthy, active stem cells can have systemic effects on aging throughout the entire body. When stem cells are functioning optimally, they don't just repair local damage, they also release beneficial factors that can improve the health and function of nearby tissues. This discovery suggests that genetic editing of stem cells might have benefits far beyond simply improving tissue repair. Enhanced stem cells might serve as cellular fountains of youth, continuously releasing rejuvenating factors that help maintain the vitality of your entire body. They could serve as biological pacemakers, setting a rhythm of renewal that keeps all your tissues functioning in harmony. As you rest in this peaceful moment, feeling the gentle rhythm of your breath and the quiet beating of your heart, take a moment to appreciate the remarkable fact that within you right now, millions of stem cells are quietly maintaining the possibility of renewal. They are your body's promise to itself that repair is always possible, that regeneration never completely stops, that the fountain of youth flows not from some distant spring, but from the very cells that make you who you are. These sleeping seeds of renewal carry within themselves the potential to bloom into whatever healing your body might need. And now, with the advent of genetic editing technologies, we stand at the threshold of being able to tend this internal garden with unprecedented care, ensuring that your fountain of youth continues to flow with the vitality of springtime, even as the seasons of life continue their ancient dance around the question of what it means to grow older while remaining perpetually capable of growing new. Imagine walking through an ancient forest, where some leaves have forgotten how to fall. They cling to branches long after their season has passed, no longer green and vibrant, but brown and weathered, blocking sunlight from reaching the new growth below. This is happening right now, in the microscopic forest of your body, where some of your cells have learned to ignore one of nature's most fundamental lessons when it is time to let go. Your body is home to trillions of cells, each one following an intricate dance of birth, growth, service, and graceful departure. Most cells live their lives like perfect seasons, knowing instinctively when their work is done, when their DNA has accumulated too much damage, when their machinery has grown too weary to function properly. When this moment arrives, they activate an elegant program called apoptosis, a word that sounds like poetry and means, quite literally, the falling away of petals. But some cells, as they age and accumulate damage, choose a different path. Instead of gracefully exiting the stage, they enter a state called senescence, a twilight existence where they neither fully live nor completely die. Like stubborn actors who refuse to leave when the play has ended, they remain in your tissues, no longer performing their original roles but continuing to take up space and resources. You might think these senescent cells are simply harmless remnants, like old furniture in an attic, but they are far more active than their dormant appearance suggests. These cellular rebels begin secreting a cocktail of inflammatory molecules, chemicals that whisper danger to their neighbors, creating a chronic state of alarm throughout your tissues. Scientists call this the senescence-associated secretory phenotype, or SASP, and it transforms these old cells into biological troublemakers. Picture a neighborhood where a few houses have been abandoned but their sprinkler systems are broken, constantly flooding the street. The water damage spreads to nearby homes, weakening foundations, encouraging mold, making the whole area less habitable. This is what senescent cells do to your body. They leak inflammatory signals that damage healthy neighboring cells, accelerate aging, and contribute to age-related diseases. As you lie here tonight, millions of these cellular refugees are scattered throughout your tissues. In your skin, they contribute to wrinkles and reduced elasticity. In your joints, they promote arthritis. In your blood vessels, they encourage atherosclerosis. In your brain, they may play a role in neurodegenerative diseases. They are the autumn leaves that refuse to fall, creating a biological winter that never quite gives way to spring. But here's where the story becomes hopeful. Like the first warm breeze that finally convinces those stubborn leaves to release their grip, scientists have discovered that senescent cells, despite their refusal to die naturally, are not immortal. They have vulnerabilities, molecular Achilles heels that can be targeted with remarkable precision. Research laboratories around the world, or developing what they call senolytic drugs, medicines that can identify senescent cells and gently encourage them to complete their interrupted journey toward apoptosis. The word senolytic comes from the Greek, meaning to dissolve the old, and these compounds work like molecular gardeners, pruning away the dead growth to make room for new life. Early experiments in mice have been breathtaking in their results. When researchers cleared senescent cells from aging animals, the mice experienced what could only be described as a reversal of biological time. Their fur regrew thicker and shinier, their bones became stronger, their cardiovascular health improved, their cognitive function sharpened. It was as if removing those stubborn autumn leaves had allowed an entire ecosystem to regenerate. The mechanisms behind this cellular housekeeping are elegant in their simplicity. Senescent cells rely on certain survival pathways to maintain their twilight existence, molecular life support systems that keep them from completing their natural death program. Senolytic drugs work by temporarily disrupting these pathways, removing the artificial supports, and allowing these cells to follow their original programming toward peaceful departure. One of the most promising approaches targets the BCL2 family of proteins, molecular guardians that normally protect healthy cells from premature death but become corrupted in senescent cells, helping them cling to their unwanted existence. By briefly interfering with these guardians, scientists can restore the natural balance between cellular life and death. Another strategy focuses on the cellular cleanup crews, the immune system's natural ability to identify and remove damaged cells. As we age, our immune surveillance becomes less efficient, like security guards who have grown tired and less attentive. Some experimental therapies work by reinvigorating these cellular security forces, helping them recognize and eliminate senescent cells more effectively. The beauty of this approach lies in its specificity. Unlike many medical interventions that broadly affect all cells, senolytic therapies can distinguish between healthy cells that should be preserved and senescent cells that should be removed. It's precision medicine at the cellular level, strikes against aging rather than carpet bombing. But perhaps the most intriguing aspect of senescent cell research is what it reveals about the nature of aging itself. For decades, scientists viewed aging as an inevitable accumulation of random damage, like rust slowly consuming metal. The discovery of senescent cells suggests that aging might be more like a garden that simply needs better maintenance, not fundamental decay, but the manageable challenge of removing weeds that prevent healthy growth. Clinical trials in humans are now underway, testing whether the remarkable rejuvenation seen in laboratory mice can translate to human biology. Early results suggest that clearing senescent cells can improve physical function, reduce inflammation, and enhance quality of life in older adults. The first generation of senolytic drugs is already moving from laboratory benches to pharmacy shelves. As your breathing deepens and your mind begins to drift towards sleep, consider the profound implications of this cellular revolution. Your body possesses an innate wisdom about renewal and regeneration, patterns written into your very DNA. The challenge has never been about conquering death, but about removing the obstacles that prevent life from expressing its full potential. Tonight, as your cells perform their nightly maintenance and repair, imagine a future where the stubborn leaves finally learn to fall, where cellular neighborhoods remain vibrant and healthy, where the biological seasons flow as they were meant to flow. But even as we learn to clear away the old, we must also understand how to strengthen and enhance the natural repair systems that build us anew each day. As you rest in this gentle moment between waking and dreaming, let your imagination carry you to the laboratories where scientists have become students of Earth's most remarkable teachers. creatures that have mastered the art of extending life in ways that would seem magical if they weren't so beautifully real. These are the longevity masters of our planet, each one conducting its own unique symphony of genes that allows it to dance with time in extraordinary ways. In a quiet corner of the Mediterranean Sea, a small jellyfish no larger than your fingernail performs one of the most astonishing feats in all of biology. Torritopsis dohrnii, known as the immortal jellyfish, has discovered something that has eluded every other complex creature on Earth: how to reverse the aging process completely when this remarkable organism reaches the end of its natural life cycle. Instead of dying, it simply transforms back into its juvenile form, effectively becoming young again. Imagine watching this transformation unfold in slow motion, like a film played in reverse. The mature jellyfish begins to shrink, its complex adult structures dissolving away until it becomes nothing more than a small cluster of cells. But this is not death, it is rebirth. These cells reorganize themselves into a polyp, the juvenile, the form of the jellyfish, and begin growing again with all the vigor of youth. The immortal jellyfish has essentially learned to reset its biological clock to zero, over and over again, potentially forever. The genetic orchestra that conducts this incredible transformation involves the precise activation and silencing of hundreds of genes. Scientists have discovered that the jellyfish possesses genetic switches that can completely reprogram its cellular identity, allowing adult cells to become juvenile cells through a process called transdifferentiation. This is like having a symphony where the violins can suddenly become cellos and the cellos can transform into flutes, all while the music continues playing. But the immortal jellyfish is not alone in its mastery of time. Deep in the forests of California bristlecone, pine trees have been conducting their own longevity symphony for thousands of years. Some of these ancient trees began growing when the pyramids of Egypt were still young, and they continue to thrive today. Their genetic orchestras playing the same life-sustaining melodies they have been performing for millennia. The secret of these ancient trees lies in their remarkable ability to maintain their stem cells in a state of perpetual youth. While most organisms experience a gradual decline in stem cell function as they age, bristlecone pines have evolved genetic mechanisms that keep their growth-producing cells as vigorous as they were when the tree was a seedling. Their DNA repair systems work with extraordinary efficiency, fixing damage almost as quickly as it occurs and maintaining the integrity of their genetic code across centuries. In the microscopic world, perhaps the most studied longevity master is a tiny worm called, Habitus elegans. This humble creature, no larger than a comma on this page, has taught scientists more about the genetics of aging than almost any other organism. Through careful genetic manipulation, researchers have discovered that changing just a single gene in these worms can double or even triple their lifespan, allowing them to live the equivalent of 200 human years while remaining healthy and active. The genetic pathways that control aging in these worms are remarkably similar to those found in humans. They involve genes that regulate cellular stress responses, DNA repair mechanisms, and the efficiency of cellular energy production. When these pathways are optimized through genetic modification, the worms don't just live longer, they live better, maintaining their youthful vigor and mobility throughout their extended lives. Scientists have identified several key genetic themes that run through the longevity symphonies of all these remarkable creatures. One of the most important is the insulin-like growth factor pathway, which controls how cells respond to nutrients and stress. In long-lived organisms, this pathway is often fine-tuned to promote cellular maintenance and repair rather than rapid growth and reproduction. It's as if these creatures have learned to invest their biological resources in preservation rather than expansion. Another crucial theme is the sirtuin gene family, which produces proteins that act like cellular housekeepers, maintaining the organization and function of cells even under stressful conditions. These genes are highly active in long-lived species and seem to coordinate many of the cellular processes that prevent aging. Some scientists refer to sirtuins as longevity genes because of their powerful effects on lifespan and halterspan. The study of exceptionally long-lived humans has revealed similar genetic themes. Centenarians, people who live to be 100 or older, often carry genetic variants that enhance their DNA repair capabilities, improve their resistance to cellular stress, and optimize their immune system function. These genetic advantages don't necessarily prevent all diseases, but they seem to delay the onset of age-related conditions and help maintain overall vitality into very advanced age. Perhaps most remarkably, scientists have discovered that many of the genetic factors that promote longevity can be artificially activated using the tools of modern biotechnology. Genetic editing techniques like CRISPR allow researchers to modify the expression of longevity genes in laboratory animals, often with dramatic effects on both lifespan and health span. Mice with enhanced DNA repair genes live longer and remain healthier as they age. Flies with modified sirtuin genes show improved resistance to cellular stress and extended lifespans. The prospect of applying these discoveries to human longevity is both exciting and complex. Scientists are working to understand exactly how the genetic orchestras of longevity work. mapping out the intricate relationships between different genes and their effects on aging. This research has revealed that longevity is not controlled by a single genetic switch, but rather by networks of genes working together in harmony, like instruments in a vast biological symphony. Some of the most promising research involves studying the genetic profiles of exceptionally long-lived families. These families often share genetic variants that seem to provide protection against age-related diseases and promote healthy aging. By understanding these protective genetic factors, scientists hope to develop therapies that could provide similar benefits to people who weren't born with these natural advantages. The research has also revealed the importance of epigenetic factors, modifications to DNA that can turn genes on or off without changing the underlying genetic code. These epigenetic marks can be influenced by lifestyle factors like diet, exercise, and stress management, but they can also be modified through genetic editing techniques. This opens up the possibility of not just inheriting longevity genes, but actively programming them into our cells. One of the most intriguing discoveries is that the genetic factors that promote longevity often work by enhancing the body's natural maintenance and repair systems. Long-lived organisms don't avoid cellular damage, they simply repair it more efficiently. Their DNA repair mechanisms work more effectively, their cellular waste removal systems function more efficiently, and their stem cells remain more active throughout their lives. This insight has led scientists to focus on developing interventions that could enhance these natural repair systems in humans. Genetic editing could potentially optimize the genes that control DNA repair, cellular housekeeping, and stem cell function, effectively giving our cells the same longevity advantages that nature has provided to the immortal jellyfish, ancient trees, and long-lived worms. The research into longevity genetics has also revealed the remarkable plasticity of the aging process. Aging is not a fixed, unchangeable program written into our genes. Instead, it is a dynamic process that can be influenced by genetic factors, environmental conditions, and potentially by therapeutic interventions. The genetic orchestras of longevity are not playing predetermined songs, they are improvisational jazz ensembles, capable of adapting their music to changing conditions. As you drift deeper into the peaceful realm of sleep, imagine the genetic orchestra playing softly within your own cells. Each gene is like an instrument, contributing its unique voice to the symphony of your existence. Some instruments play the melodies of growth and repair. Others provide the rhythmic foundation of cellular maintenance, and still others contribute the harmonies that keep everything working in perfect coordination. The creatures that have mastered longevity have not discovered some secret magic, they have simply learned to conduct their genetic orchestras with exceptional skill, keeping all the instruments in tune and all the sections playing in harmony. And now, for the first time in human history, we are beginning to understand how to conduct our own genetic symphonies, potentially extending the beautiful music of life far beyond what previous generations could have imagined possible. But as we stand at the threshold of this remarkable possibility, we must carefully consider the profound questions that arise when we contemplate the power to extend human life. Dramatically, questions that touch the very heart of what it means to be human and how we should use such transformative capabilities. In the quiet sanctuary of this moment, as your breath moves in and out with the gentle rhythm of rest, let us pause at the threshold of possibility and listen to the whispered questions that arise when we contemplate the power to extend human life beyond its natural boundaries. These are not the harsh debates of politics or the cold calculations of economics, but the gentle, necessary conversations we must have with our own hearts. As we stand at the edge of potentially rewriting the very meaning of human existence. Imagine, for a moment, that tomorrow scientists announced they had perfected the genetic editing techniques needed to slow aging dramatically, potentially allowing humans to live healthy, vigorous lives for 2, 3, or even 5 centuries. This is not science fiction. It is a possibility that grows more realistic with each passing year as our understanding of longevity, genetics deepens and our editing tools become more precise. But with this extraordinary possibility comes questions that have never been asked before in human history. What would it mean to live for 300 years? How would such an extended lifespan change the way you experience relationships, career, learning, and personal growth? Consider the profound implications of having not just one lifetime to explore your potential, but several lifetimes contained within a single continuous existence. You could master multiple careers, learn dozens of languages, travel to every corner of the earth, and still have centuries left to discover new passions and purposes. Yet even as we marvel at these possibilities, deeper questions emerge like gentle currents beneath the surface. If some people could live for centuries while others lived normal lifespans, would we be creating a world divided not just by wealth or status, but by time itself? Would those with extended lives become a separate class of humanity, carrying forward centuries of accumulated knowledge, experience, and resources while others lived their brief, traditional lifespans? The whisper of fairness speaks softly but persistently in these considerations. Throughout human history, death has been the great equalizer, the one experience that unites all people regardless of their circumstances. Rich and poor, powerful and powerless, wise and simple all have faced the same fundamental limitation of a finite lifespan. If we remove this shared constraint, we might be creating inequalities more profound than any that have existed before. Perhaps the question of fairness is more complex than it first appears. Consider the moral argument that if we possess the knowledge and capability to prevent suffering and extend healthy life, do we not have an obligation to do so? When we develop treatments for diseases, we don't withhold them because not everyone can access them immediately. Instead, we work to make them available as widely as possible, understanding that new technologies often begin with limited access before becoming more broadly available. The economics of longevity present their own gentle but persistent questions. How would society function if retirement at 65 became retirement at 265? What would happen to career progression, leadership succession, and the natural cycles of generational change that have driven human progress? Would innovation slow if positions of influence were held by the same individuals for centuries? Or would the accumulated wisdom and experience of extended lives accelerate human advancement in ways we can barely imagine? There is also the whispered question of psychological well-being. The human mind has evolved to cope with the psychological realities of a lifespan measured in decades, not centuries. Would extended longevity bring extended happiness and fulfillment, or might it lead to new forms of psychological challenges that we have never encountered? How would identity and personal growth unfold across centuries rather than decades? Would the intensities of love, loss, hope, and dream maintain their poignancy across such extended timescales? The environmental implications of dramatically extended human lifespans speak in their own quiet but urgent voice. Our planet already strains to support the current human population with our current lifespans. What would be the ecological impact of billions of people living for centuries instead of decades? Would we need to fundamentally reimagine human reproduction, career cycles, and resource consumption to create a sustainable model for dramatically extended life? Yet perhaps these concerns, valid as they are, reflect our current limitations rather than insurmountable obstacles. Throughout history, humans have shown remarkable adaptability in the face of technological and social changes. The same innovative capacity that might allow us to edit away aging could also help us solve the social, economic, and environmental challenges that extended longevity would create. Consider the possibility that extended lifespans might actually enhance human wisdom and environmental stewardship. People who expect to live for centuries might make very different decisions about sustainability and long-term planning than those who expect to live for decades. The political and economic systems that govern our world might become more focused on long-term outcomes if the decision-makers themselves expected to experience the consequences of their choices for hundreds of years. There is also the gentle question of what we might lose in gaining so much time. Would the urgency that drives human creativity and achievement diminish if we had centuries to accomplish our goals? Does the knowledge of our mortality give meaning and intensity to our experiences that might be diluted across extended lifespans? Or would extended life simply provide more opportunities for meaning, creativity, and profound experiences? The question of consent whispers through these considerations as well. Future generations will inherit whatever decisions we make about longevity technologies. Are we entitled to make choices that will fundamentally alter the human experience for all who come after us? Yet the same question applies. In reverse, do we have the right to withhold potentially transformative benefits from future generations because of our own uncertainties? Perhaps most profoundly, we must ask whether extending human lifespan represents an improvement in the human condition or a departure from our essential humanity. Are the cycles of birth, growth, aging, and death fundamental to what makes us human, or are they simply biological constraints that technology can appropriately address? Would humans who live for centuries be better versions of ourselves, or would they be something fundamentally different? These questions do not have easy answers, and perhaps that is as it should be. The power to dramatically extend human life is not a power to be wielded lightly or quickly. It requires careful thought, extensive dialogue, and the kind of wisdom that emerges from considering not just what we can do, but what we should do. The ethical whispers of longevity remind us that scientific capability alone is not sufficient justification for action. We must also consider the broader implications for human society, for our relationships with each other and with the natural world, and for the meaning and purpose of human existence itself. As you rest in this moment of contemplation, feeling the gentle rhythm of your own mortality in each heartbeat and breath, consider that these questions are not distant philosophical puzzles. They are immediate, practical considerations that our generation may need to address. The choices we make about longevity research and its applications will shape the future of human experience in ways that may reverberate for centuries. The whispered conversations we have with our own hearts about these possibilities are not separate from the scientific research itself. They are an essential part of it. The development of longevity technologies must be guided not just by what is scientifically possible, but by what is ethically wise and socially beneficial. And so we find ourselves poised at this remarkable moment in history, holding in our hands the potential to transform the human experience of time itself. while simultaneously carrying the responsibility to use this power with profound wisdom and care as we step forward into tomorrow's uncharted possibilities. As you settle into the deepest layers of rest, let your mind drift forward through time, to glimpse the tomorrow that scientists and researchers are quietly building in laboratories around the world. This is not a distant future measured in centuries, but a tomorrow that may arrive within your own lifetime, a time when the genetic editing of aging might transition from experimental possibility to practical reality, fundamentally altering what it means to grow older. In research facilities spanning the globe, the first tentative steps toward this future are already being taken. Clinical trials are beginning to explore whether the genetic modifications that extend lifespan in laboratory animals can be safely and effectively applied to humans. These early studies move with the careful precision of explorers mapping unknown territory, testing each step before moving forward. Guided by both scientific rigor and ethical responsibility, picture the researchers working in these laboratories, their days filled with the meticulous work of translating decades of basic research into practical treatments. They culture human cells in carefully controlled environments, editing genes with molecular precision and observing how these modifications affect cellular aging processes. Cell cultures that once would have died after a few dozen divisions now continue growing and dividing, maintaining the vigor of youth far longer than nature intended. The first human applications of anti-aging genetic editing are likely to be modest and carefully targeted, rather than attempting to dramatically extend overall lifespan immediately. Researchers are focusing on editing specific genes that contribute to age-related diseases. Early trials might involve editing genes that affect cardiovascular health, cognitive function, or immune system aging, targeting the conditions that most significantly impact quality of life as we age. Imagine receiving a treatment that edits the genes controlling your telomerase enzyme, helping your cells maintain their protective chromosome caps and potentially slowing cellular aging throughout your body. Or consider a therapy that optimizes the genes responsible for your cellular repair mechanisms, allowing your body to maintain itself with the efficiency of a much younger person while your chronological age continues to advance. These early treatments might not make you immortal, but they could extend your healthy lifespan significantly, perhaps adding decades of vigorous disease-free life to your natural span. Instead of experiencing the gradual decline typically associated with aging, you might maintain the physical and mental capabilities of middle age well into what we now consider old age. The delivery methods for these genetic modifications are becoming increasingly sophisticated and less invasive, rather than requiring complex surgical procedures. Future anti-aging treatments might be delivered through simple injections or even oral medications. Scientists are developing ways to package genetic editing tools in tiny capsules that can be targeted to specific tissues or organs, allowing precise modifications without affecting the rest of your body. One promising approach involves using modified viruses as delivery vehicles for genetic editing tools. These viruses are engineered to be completely harmless while carrying the CRISPR components needed to edit specific genes. When injected into your bloodstream, they could travel to target tissues and perform precise genetic modifications that slow aging processes in your heart, brain, muscles, or other organs. The timeline for these developments is accelerating rapidly. What once required decades of research can now be accomplished in years or even months thanks to advances in genetic editing, technology, and computational biology. Artificial intelligence is helping researchers identify the most promising genetic targets and design more effective treatments. Automated laboratory systems can test thousands of genetic modifications simultaneously, dramatically speeding the pace of discovery. Within the next decade, we may see the first approved therapies that use genetic editing to address specific aspects of aging. These treatments might initially be available only for serious age-related conditions, but as their safety and effectiveness are established, they could become more broadly accessible for general longevity enhancement. The regulatory pathways for these treatments are still being established. Government agencies around the world are grappling with how to evaluate and approve therapies that don't just treat diseases but potentially extend healthy human lifespan. This regulatory landscape will likely evolve rapidly as the science advances and public demand for these treatments grows. Consider how this tomorrow might unfold in your own life. Perhaps at age 60, you might receive a genetic treatment that optimizes your cellular repair mechanisms, allowing you to maintain the physical capabilities of a 40-year-old at 80. Another treatment might refresh your immune system, keeping it functioning like that of a younger person. At 120, you might still be living independently, pursuing new interests and maintaining relationships with people across multiple generations. The social implications of this tomorrow are as profound as the scientific ones. Educational systems might need to adapt to serve learners who could be acquiring new skills and knowledge for centuries rather than decades. Career paths might become more fluid and diverse as people have time to master multiple professions. Relationships might develop new forms and rhythms as couples potentially share centuries together rather than decades. Economic systems would likely undergo fundamental transformations. The concept of retirement might become obsolete or be completely reimagined. Investment strategies, insurance models, and social support systems would need to adapt to lifespans that could extend for centuries. The economic value of human experience and accumulated knowledge might increase dramatically as individuals have more time to develop expertise and wisdom. Healthcare systems would shift from treating age-related diseases to preventing them through genetic optimization, rather than managing the decline associated with aging. Medical professionals might focus on maintaining optimal function throughout extended lifespans. Preventive genetic editing could become as routine as current preventive healthcare measures like vaccinations or health screenings. The research community is already preparing for this future by establishing ethical frameworks and safety protocols for anti-aging genetic editing. International collaborations are being formed to share research findings and establish common standards for safety and efficacy. These preparations are essential for ensuring that the transition to dramatically extended lifespans happens smoothly and equitably. One of the most intriguing aspects of this tomorrow is how it might change human relationships with time itself. If you knew you could live for centuries, how would you approach learning, relationships, and personal growth? Would you take more risks, knowing you had more time to recover from setbacks? Would you be more patient with long-term projects and relationships? Would you develop deeper appreciation for the present moment, or would extended time create new forms of temporal anxiety? The psychological adaptation to dramatically extended lifespans represents one of the most fascinating unknowns of this tomorrow. Human psychology has evolved around the assumption of finite lifespans measured in decades. How would our minds adapt to the possibility of living for centuries? Would we develop new forms of wisdom and emotional intelligence? Or would we face unprecedented psychological challenges? Cultural and spiritual traditions would likely evolve in response to dramatically extended lifespans. Religious and philosophical frameworks that have provided meaning and purpose within traditional lifespans might need to be reimagined for extended existence. New forms of art, literature, and cultural expression might emerge to capture the unique experiences of living across centuries. The environmental implications of this tomorrow are also being carefully considered. Researchers are exploring how dramatically extended human lifespans could be made sustainable within Earth's ecological limits. This might require fundamental changes in how we approach reproduction, resource consumption, and environmental stewardship. The same innovative capacity that enables life extension might also need to be applied to creating sustainable models for extended human existence. As you drift deeper into sleep, carrying with you these images of tomorrow's possibilities, consider that this future is not fixed or predetermined. It is being shaped by the choices and actions of researchers, policymakers, and citizens today. The tomorrow we might touch is not something that will simply happen to us. It is something we are actively creating through our collective decisions about how to develop and apply these powerful technologies. The tomorrow where aging becomes optional might be closer than we think, approaching with the quiet inevitability of dawn breaking over a landscape that has been transformed by the gentle but persistent work of those who dare to imagine that the fundamental constraints of human existence might not be as fixed as we once believed, opening doors to possibilities that invite us to contemplate not just how long we might live, but how fully we might embrace the gift of extended time. As you rest in this peaceful space between waking and sleeping, having journeyed with me through the remarkable possibilities of genetic editing and life extension, let us pause to reflect on a profound paradox. Perhaps the greatest gift that comes from exploring how we might extend life dramatically is discovering the exquisite beauty and value of the time we already have, whether it spans decades or centuries. Throughout our exploration of CRISPR, telomeres, stem cells, and the genetic orchestras of longevity, we have marveled at the possibility of extending human life far beyond its current boundaries. We have contemplated futures where aging might become optional, where centuries could unfold before us filled with experiences, relationships, and discoveries. Yet, in contemplating these extended tomorrows, we might have discovered something even more precious, a deeper appreciation for the present moment, and the inherent value of our temporary existence. Consider the gentle irony that in seeking to extend time, we learn to treasure it more completely. When we understand the intricate cellular mechanisms that count away our days, the shortening telomeres, the accumulating genetic changes, the gradual decline of our repair systems, we begin to see each moment not as an inexorable step toward an end, but as a miracle of coordinated molecular activity that makes consciousness, love, creativity, and wonder possible. The research into longevity has revealed something beautiful about the nature of life itself. It is not the length of existence that creates meaning, but the depth of experience within whatever time we have. The immortal jellyfish, despite its ability to reset its biological clock indefinitely, lives each cycle of its existence with the same simple intensity as creatures with much shorter lifespans. The ancient bristlecone pines grow with the same patient determination whether they live for decades or millennia. This understanding offers a profound form of wisdom. Whether we live for 80 years or 800 years, each moment carries the same potential for beauty, connection, and meaning. A sunset viewed at age 25 is no less magnificent than one viewed at age 250. The love shared between friends remains equally precious, whether it spans decades or centuries. The joy of learning something new, creating something beautiful, or helping another person carries the same intrinsic value regardless of how much time stretches before or behind us. In contemplating the possibility of dramatically extended lifespans, we are also invited to consider what truly matters in the time we have. If you knew you might live for 300 years, would you spend your days differently than you do now? Would you be more patient with difficult relationships, knowing you had more time to work through challenges? Would you pursue deeper learning, make bolder creative choices, or invest more fully in long-term projects and commitments? Perhaps more importantly, would you appreciate the simple, everyday miracles more fully? The warmth of morning sunlight streaming through your window, the taste of fresh water when you're thirsty, the comfort of breathing easily, the gentle rhythm of your heartbeats. These fundamental experiences remain equally precious. Whether they occurred during the 70th year of your life or the 270th, the exploration of genetic editing and aging has also revealed the remarkable wisdom already present in the natural cycles of life. The processes that cause us to age are not simply failures or flaws in our biological design. They are often trade-offs that serve important purposes in the grand scheme of life on Earth. The cellular mechanisms that limit our lifespans also protect us from cancer. The genetic changes that accumulate over time contribute to the diversity that drives evolution and adaptation. This doesn't mean we should not pursue the extension of healthy human lifespan, quite the contrary, but it suggests that we should approach these possibilities with humility and respect for the profound complexity of life itself. The genetic editing tools that might allow us to live longer also remind us of the extraordinary intricacy and beauty of the biological systems that sustain us each day. There is also deep wisdom in accepting that uncertainty and change are fundamental aspects of existence regardless of how long we live. Even if we could extend human lifespans dramatically, we would still face the unpredictability of life, the challenges of growth and change, and the fundamental mysteries of consciousness and existence. Extended longevity might give us more time to explore these mysteries, but it wouldn't eliminate them. In fact, the prospect of living for centuries might make the acceptance of uncertainty even more important. Over extended lifespans, we would likely face more changes, challenges, and transitions than we can currently imagine. The psychological and spiritual skills needed to navigate change gracefully enters adaptability, resilience, and the ability to find meaning in difficult circumstances might become even more crucial in extended lives. The wisdom of accepting time also includes accepting our interdependence with others and with the natural world around us. Whether we live for decades or centuries, we remain part of larger systems and communities that support and sustain us. Extended lifespans would not make us independent of these relationships. They would likely make us more deeply aware of how interconnected we truly are. This awareness might cultivate a different kind of responsibility toward future generations and the environment. If we expected to live for centuries, we might become more invested in creating sustainable systems and solving long-term challenges. The knowledge that we would personally experience the consequences of today's environmental and social decisions might motivate us to act with greater wisdom and foresight. The contemplation of dramatically extended lifespans also invites us to consider the cyclical nature of growth and renewal that operates at many levels of existence. Even with genetic editing, our extended lives would likely involve cycles of growth, maturity, rest, and renewal. We might experience multiple career cycles, relationship phases, and periods of personal transformation, each offering opportunities for learning and development. Perhaps the deepest wisdom that emerges from exploring life extension is the recognition that meaning and fulfillment don't depend primarily on the quantity of time available to us, but on how fully we inhabit whatever time we have. A life lived with presence, compassion, curiosity, and connection creates profound value, whether it spans 70 years or 700 years. This doesn't diminish the importance of the scientific research into aging and longevity. Extending healthy human lifespan could reduce enormous amounts of suffering and create unprecedented opportunities for human flourishing. But it reminds us that the ultimate value of extended life would come not from its duration, but from what we choose to do with the additional time we might be given. As you drift deeper into the restorative embrace of sleep, carrying with you all that we have explored together, let yourself feel gratitude for this present moment, for the extraordinary complexity of cellular processes that sustain your life, for the genetic inheritance that connects you to billions of years of evolutionary history, and for the remarkable fact that you exist at this particular moment in cosmic time, when humans are just beginning to understand and potentially edit the fundamental mechanisms of aging. Whether the future brings dramatically extended lifespans or not, you are living right now in the midst of one of the most extraordinary periods in the history of life on Earth. You are witnessing and participating in humanity's first attempts to consciously direct its own biological evolution. This moment itself is precious and unrepeatable, worthy of wonder and appreciation regardless of how many more moments might follow. The greatest wisdom may be to hold both possibilities with equal grace, to pursue the extension of healthy human life with dedication and hope, while simultaneously embracing the profound beauty and meaning available in whatever time we have. In doing so, we honor both the magnificent complexity of life as it is and the breathtaking possibilities of what it might become. Finding peace in the knowledge that each moment, however brief or extended, contains within itself the potential for infinite value and meaning. Description: the hidden clocks inside you, the secret dance of DNA and aging. You're not aging by accident, you're following a silent, ancient rhythm written deep within your cells. This video invites you into the surreal, microscopic world where time doesn't tick, it pulses. Here, you'll awaken to a truth hidden in your very biology. Your identity, your energy, even your aging, are governed by invisible clocks ticking quietly beneath your awareness. The journey ahead isn't just scientific, it's spiritual. It's a meditation on time, entropy, and the fragile miracle of being alive. Through introspective narration and slow science visuals, we'll explore the metaphysical poetry of your biology, shadow work at the cellular level. This is philosophical healing for the body and mind, revealing how dual memory and identity emerge from the molecular library within. What you'll gain in this slow breath, deep thought episode: a poetic look into the process of cellular aging, the mystery of DNA as a living library, how your body rebuilds itself every moment, insights into entropy, time and molecular imperfection, the science and symbolism of self-renewal, hope from organisms that reverse aging, reflections on mortality, memory, and meaning, the possibility of rewriting your biological future. If this video moved you in silence, let it echo. Like, share, and subscribe. Your body is listening. Your soul is remembering. Tags: molecular timekeepers, DNA and aging, emotional storytelling, science, philosophical biology, genetic code explained, introspective science video, poetic DNA narration, biological clocks ticking, slow science visuals, sleep, science, meditation, entropy and aging, self-renewal process, cellular aging explained, genetic imperfections, spiritual biology, storytelling about DNA, deep science, philosophy, mitochondria, energy decline, psychological aging process, gentle science voiceover, body as a city metaphor, philosophical health journey, science of renewal, dreamlike science narration, inner universe exploration, philosophical healing content, reverse aging discoveries, emotional science narration, existential science journey, shadow work biology. I am Ajasin, the hush between heartbeats. A universe of time begins to speak. Ancient clocks pulse beneath your skin, counting moments your mind can't remember. A single breath carries the wisdom of a billion. Silent cells. Your body becomes a city that never sleeps, its citizens building life in the dark. In every cell, a secret timekeeper whispers the rhythm of your becoming. Molecules dance to a song composed before the first sunrise. Time doesn't tick, it blooms and fades in chemical wavesa. Newborn's softness is slowly sculpted into the wisdom of weathered hands. Proteins assemble like midnight architects, stitching you back together while you dream. The guardians of your body stand watch, unseen soldiers in a war you never knew was waged. A single error in the script of life can echo louder than 1,000 perfect lines, like candlelight on parchment. Your cells copy themselves with trembling reverence. Your DNA is an ancient manuscript, fragile, flawed, and full of forgotten. Versus aging is the poetry of entropy, graceful, inevitable, and deeply human. The mitochondria dim their lanterns, casting long shadows across your thoughts. Time doesn't steal youth, it trades it for clarity. You are not decaying, you are translating yourself into something truer. Each wrinkle is a line of music in the symphony of becoming. The river that is you flows onward, always changing, never gone. Beneath every cell division, the ghost of a memory lingers. Your body sheds itself like a snake, but never fully forgets its skin in the marrow of your bones. Construction crews rebuild what time has weathered. Renewal is not resistance, it is surrender to the eternal cycle. You are a masterpiece in motion, repainted with every breath, even as you age. You are an act of daily resurrection. A stone step wears down not in one blow, but through a million gentle passings. The blueprint of you evolves with every whispered imperfection. The dance of DNA is both flawed and divine, sacred in its repetition. Your genetic code is a scroll, read aloud by molecular monks in a silent cathedral. In the quiet library of your cells, history is rewritten in invisible ink. 4 letters spin an epic older than myth, newer than dawn. The most intimate autobiography is written in a language no tongue can pronounce. The double helix unfurls like a secret never meant to be kept. Molecules trace the spiral staircase of your being, translating dust into destiny. Protein factories hum like choirs, assembling miracles in total silence. Each enzyme moves with the grace of a calligrapher scribing the soul. The rhythm of replication is a sacred ritual, often unnoticed, never unfelt. One misplaced letter can break a body or awaken a transformation. Mutation is the breath of evolution and the sigh of mortality. Somewhere in your cells, Time forgets its rules and life rewrites its terms. Jellyfish and trees guard the keys to a clock we still don't understand. Time can be bent, reversed, or paused but never silenced. Your cells listen for forgotten instructions buried in the genome's ancient script. Aging is not failure. It's a translation of existence into wisdom. The DNA that shaped your first breath still sings beneath your skin. To exist is to be rewritten, not once, but endlessly. The universe folded you into form and now teaches you to unfold in the vast library of your body. The smallest page holds the most sacred truth. Even now your cells are a quiet dance of rebirth. What you call aging is really the story of your becoming, written in molecular ink.